Thank you for joining me in this module entitled Drainage Water Quality Monitoring Overview. This is a high-level overview of why and how we monitor drainage water quality. We'll also discuss different experimental designs for different drainage study objectives, and I hope you'll increase your familiarity with data quality and uncertainty around monitored data. We'll start with, why is edge-of-field tile drainage monitoring important, and why should we do this? I should make clear from the start of this module that our context is subsurface drainage. There's a lot of overlap between concepts and methods for different applications of water monitoring, like stormwater monitoring or wastewater monitoring, but we're focused on subsurface drainage. With that in mind, the primary purposes of edge-of-field monitoring are to both understand the magnitude and extent of nutrient loss through tile drains, and to assess if our conservation practices are working. In other words, we use edge-of-field monitoring to determine if the practices that we promote for reducing nutrient loss through tile drains are working like we think that they should. For agronomists and environmental scientists and soil scientists, in-field monitoring would include soil sampling, gas sampling, biomass sampling, yield monitoring, and other things. For researchers focused on water, edge-of-field sampling is an assessment of water that is leaving the field. As you would guess, this monitoring is necessarily done literally at the edge of the field, or in some cases at the edge of a plot if we're doing plot scale research, which I talk about later on in this module. That's not to say water people aren't interested in the in-field water quality or quantity. We do often use monitoring wells, piezometers, lysimeters, tensiometers, etc. to evaluate water movement and water quality in the field. But this module focuses on edge-of-field monitoring. We're lucky in some ways with subsurface drainage monitoring because we have a pipe to work with. Granted, it's 3 feet below ground, so we do have to figure out how to access the pipe, but nevertheless, our water is generally contained. Monitoring surface runoff tends to be much trickier because surface runoff always finds the path of least resistance to leave the field. Typically, flumes are required if you want to measure and monitor surface runoff. Here's another example of the infrastructure required to measure surface runoff from plots. But again, while we're not talking about surface runoff in this video, many of the same concepts apply in terms of water monitoring. Let's talk a little more specifically about what we are actually looking for in this type of drainage monitoring. Starting in the upper left corner, we need to know, or to monitor, the concentration of the pollutant of interest in the water. In tile drainage monitoring, these pollutants are often nutrients like nitrate, nitrogen, or phosphorus. We monitor nutrient concentrations most generally by collecting a sample of the water and analyzing that water sample for nutrients in a lab. If you're actually going to be doing water sampling, I highly recommend completing the next module in this series, the Let's Do It module, where we talk in depth about tile drainage water sampling. In that next module, we talk about grab sampling versus automated sampling, flow-based versus time-based samples, and proper QA/QC in the lab for sample analysis. The second part of edge-of-field tile drainage monitoring is knowing how much water leaves through the tile. But what we're really working toward here is being able to estimate nutrient loads and losses, not just simply knowing how much water leaves the field. The end game is being able to estimate nutrient loss, and we need to know both concentration and water volume to estimate annual nutrient losses. A little terminology here. A load is simply a mass, like 400 pounds or 400 kilograms of nitrogen flowed out of that given tile last year. A loss, on the other hand, is a mass that's normalized per area. For example, 20 pounds of nitrogen per acre flowed out of that tile last year. Loss is sometimes called yield, which probably sounds familiar because we talk about grain yield on a per acre basis, that is, bushels per acre. And this term loss should also sound familiar, at least in Illinois, because we call our Illinois nutrient strategy the Illinois Nutrient Loss Reduction Strategy. I'll take one side step here to point out that sometimes concentrations are used on their own for important things like eutrophication criteria for streams, drinking water standards like 10 milligrams per liter nitrate nitrogen, and toxicity criteria for aquatic life. All of the units in those examples were units of concentration, not load and not loss. But just to reiterate, while concentrations provide important information on their own, When we talk about edge of field tile drainage monitoring, we most generally want nutrient concentrations and flow information together. Knowing how much water leaves a field through a tile pipe involves flow monitoring. And again, if you're actually going to be doing this, I highly recommend completing that next Let's Do It module in the series where we talk in depth about tile drainage flow monitoring. That's the intro. See you next time.